So good afternoon, everybody. I have put together slides on 3 different areas. A little bit about where we are in the outbreak, some lessons from work that I have done and others have done previously on SARS animal models, and some concerns about vaccines. Indeed, I mentioned today, I really think are very important for the immunology community in Melbourne to start thinking about. I think we would all benefit from having people that come to a problem from different angles engaging on this. So do I control these? Yeah. So I'm starting with this slide many of you are familiar with. This is something that the Johns Hopkins University has put together. It's updated regularly. This was as of last night, so by no means would be accurate by midday today. But it gives you an idea on a map of the world where the confirmed cases are and they're by region. They also keep a tally of total deaths and the total number of people recovered. So you can see that what started off in January as something that was limited initially to the city of Wuhan and then to Hubei province has really become a global pandemic. Specifically in Australia, these are the data from the website at the bottom that list the number of cases, and you can see that we've had a very sort of recent but significant uptick in the number of cases. When you break it down by state and territory, in black are the data from New South Wales, followed after with a gap with Victoria in the yellowish color, and Queensland in blue, in the turquoise blue, and WA and ACT, etc. lower. So this website actually lists the number of deaths and confirmed local transmission as well. The new cases nationwide by day are in this very colorful graph, and it just indicates that the number of travel-related transmissions still dominate, but there is actually an increasing number where it's either unknown or local transmission. So what happens in the timeline of coronavirus infections, the first week tends to be quite mild, and hospital admission is typically about 7 days after the onset of symptoms. The worsening of...
Sorry, can everybody please make sure they've muted their microphones who are listening in via Zoom, because apparently there's some interference. So if everyone Zooms, then we should be okay. We can't hear it in this room, so.
There is a mute all function.
Is there a mute all function?
Under participants.
Better to know.
Go to manage participants. Up the top, manage participants.
It was there, it just disappeared. There it is.
Click on that, and there's mute all.
And then
no. Okay, all right. Okay.
okay.
Sorry.
No, that's helpful. Hopefully somebody will tell you if it's better.
I've got one person here letting me know.
Okay.
Nick Gerard.
Okay, so the timeline, so the first week tends to, people tend to have mild symptoms, and those that are going to have worsened clinical signs seems to happen in the second week. And there's actually quite a rapid progression in that second week from shortness of breath to ARDS and ICU admission. So that first week is mild, but then there's a worsening in the second week, and that progression is fast. Okay, so there's a paper that just came out a day or 2 ago from Neil Ferguson's group at Imperial College London, where they looked at the impact of non-pharmaceutical interventions, and they looked at I just picked out a few tables that I thought might be of interest, and this is by decade. The percent of symptomatic cases that their models suggest would require hospitalization, require critical care, and then the infection fatality ratio. And this really climbs very steeply after age 60, and then really goes up at age 80. So the mitigation strategies that they've talked about in this modeling is, there are several different strategies: case isolation, voluntary home quarantine, social distancing of those over 70 years of age, social distancing of the entire population, or closure of schools and universities. And on this graph, you could see the effect of each of these mitigation strategies alone and in combination. And the take-home point from this is that the black line on top, which is a very remarkable high peak, is lowered by almost any of these interventions, but the combination is better than any of them alone. So that is the bit of background that I was going to give you about the outbreak itself. And I'm now going to switch to talking about SARS. I worked on SARS for about 5 years when I was at the NIH. I had a lab in the intramural program where we developed animal models for SARS. The intent was to develop animal models and vaccines, but what happened in reality was what's happening today, and that is that many different platforms are available, and people can make vaccines without ever touching the live virus. And so what they needed, though, was the ability to test their vaccine strategies in animal models. And on this slide, I've listed the number of models that we developed in my lab. So we had a number of different mouse models. We looked at a number of different knockout mice to understand the biology. We had a hamster model, which was my favorite. And then we developed a number of non-human primate models as well. So I'll just take you through some of the key experiments. This was the first experiment that we did, and this was in young mice, young adult VALP-C mice. We administered the virus intranasally to lightly anesthetized mice, the way we would do with flu or any other respiratory virus. We tried 3 different doses, 10 to the 3 or 10 to the 5 TCID50 administered intranasally. And at serial time points, we sacrificed mice, took out their lungs or nasal turbinates, and titered the amount of virus that we recovered that was present in those tissues. And you can see that the 10 to the 3 and 10 to the 5 doses, virus replicated well in the lungs and nasal turbinates. Now, it turns out that none of these mice actually had any clinical signs of illness. There was no weight loss, no ruffled fur. They really were least bothered by the whole event, and they had cleared virus by day 7. But when we looked to confirm what was happening in the lungs, we did, in collaboration with pathologists at the CDC, we did immunohistochemistry as well as in situ hybridization, and this confirmed what we saw with the virus titration data, that there were viral antigen and viral nucleic acid present in the cells lining the large airways. The part of that, as part of that first set of experiments, we kept some of the mice alive while we were looking at their weight loss and so on. And so we led them at the end of a month and demonstrated in a neutralizing antibody assay that we developed that these mice did develop neutralizing antibody. And when we challenged these animals with virus 1 month post primary infection, they were fully protected from replication of the challenge virus compared to a mock vaccinated, mock infected group that were challenged for the first time. So this demonstrated that primary infection protected from subsequent challenge. We also took serum from the mice that had recovered from their primary infection, and we administered it by passive transfer, undiluted or diluted 1 to 10, just to get a dose response. And we took serum from previously non-infected mice, transferred them to naive mice, demonstrated the titer, neutralizing antibody titer achieved in the recipient mice, and then challenged them with live virus, and demonstrated that, in fact, passive transfer of immune serum protected naive mice from challenge virus replication. We demonstrated this in addition with a number of different human monoclonal antibodies that were made by a number of different people, Antonio Lanzavecchia, Wayne Marasco, and the Mass Public Health Lab, and so on. Now, one of the issues with the young adult mice was the absence of any clinical signs. So clearly there was virus replication, and there's a lot you can use that model for, but there was a general concern that there was no significant clinical signs. And so we did observe in the course of some other experiments that age seemed to matter, and it certainly mattered in the clinical setting. So we then developed a model in 12-month-old Vauci mice. We call them old, but apparently the National Institute of Aging does not consider a 12-month-old mouse old. And so we're qualifying that and saying that they're 12-month-old mice. And you can see in this top graph that 12-month-old mice inoculated with the exact same dose and the same virus that we used in young adult mice resulted in weight loss. So you can see this big dip in weight. They actually had higher viral replication, higher titer virus, and we saw alveolar damage in the lungs. We teased this out quite extensively over the next couple of years and could demonstrate that in this kind of funky yellow color is when the virus titer peaks, and the pneumonitis actually follows the peak of viral replication. We saw, we did a lot of work with various cytokines and chemokines and characterized what was happening with the different cell subsets. And one of the things we did also was to look to see what depleting the CD4 or CD8 or both CD4 and CD8 T cells will do in this model. And so actually Gary Lau was one of the graduates from DMI who came to my lab as a postdoc. And so these are 12 to 14-month-old Vauci mice. We gave them the depleting CD4, CD8, or both antibodies or an isotype control at day -3 and +3, and also infected them or gave them mock infection. And what I've highlighted here in this red box shows you that CD8 depletion did not actually affect viral clearance, but CD4 T cell depletion did. This is a bit of a surprise. And so this very bright green color is the day 9 data. And we tease that out further by looking between day 7 and 12 at daily time points. And we could see on the right that you can see by immunohistochemistry, you can see persistent viral antigen at day 9 in the CD4 depleted or mice depleted of both CD4 and CD8. Interestingly, we found that there was enhanced interstitial pneumonitis in the lungs of SARS-infected mice after depletion of the CD4 T cells. That's what you see here. And this was scored as well. Now switching from the mouse model to the hamster model, and I'm not going to tell you a lot about the hamsters because apparently hamsters are exotic to Australia and cannot be imported. So I will just tell you the one bit of a really valuable experiment that was done by my colleagues Peter Collins and Una Buchholz in the Laboratory of Infectious Diseases. And they used a bovine human parainfluenza virus type 3 as a vector. And they inserted into this live bovine human parainfluenza virus type 3 the various structural proteins of SARS. So it was S, M, E, or N proteins, or a combination of S, M, and E, or just M and E. And then they had a control that had no SARS insert. And the other control was SARS infection. And this is in hamsters. And what they saw was that if you vaccinated animals with any of these constructs and came back and challenged them with the live virus, you could see that the control animals had very robust, these are log to the base 10 TCID50 per gram of tissue, so virus replicates to 5 to 6 logs in the lungs and nasal turbinates of hamsters, and only the constructs that contain the spike, their spike alone or spike with MNE, provided protection in the hamsters. And those were also the only combinations, or only vaccination strategies that induced neutralizing antibody, which is shown here. We developed 3 different monkey models initially and then tested some New World species later. And in our hands, we compared rhesus, cynos, and African green monkeys, administering 10 to the 60 CID50 of virus in 1 ml intranasally and 1 ml intratracheally. This is the strategy that we've used for flu, for paraflu, for a number of different viruses in the lab that I was in. We saw no clinical signs of disease, but remember, these monkeys are sitting in a big cage in a PC3 facility. There's not a lot of activity and not a lot of room to demonstrate any clinical signs of disease. But we could sample these animals daily, both from nasal swabs or nasal lavage and tracheal lavage. And we saw that in African green monkeys, we demonstrated a more consistent and higher viral replication in the respiratory tract. The lungs on day 2 post-infection have some inflammatory changes and a lot of viral antigen by immunohistochemical staining. So how did we use these models? We used them to test the effectiveness of immunoprophylaxis and immunotherapy, as well as vaccines. As I said, even back in 2003, 2004, 2005, people had applied a number of different platforms to develop vaccines against SARS. There were inactivated vaccines where virus was inactivated with BPL, formalin, UV, or combination. There were subunit vaccines, protein vaccines, VLPs, vector vaccines, including the one I just mentioned, MVA, adeno, VSD, and rabies, DNA vaccines, and combinations. We tested human monoclonal antibodies isolated from immortalized memory B cells from a convalescent patient, from phage display libraries, and those generated in a transgenic mouse. To summarize a whole lot of work, because we evaluated 9 vaccines and 4 monoclonals in a variety of animal models, we looked at vaccines that had been generated using several different virus strains. Vaccines had been delivered IM or sub-Q to mice, hamsters, and monkeys. And the region from the spike protein residues 13 to 877 that included an important epitope from amino acids 735 to 882 was essential for an effective protein-based vaccine. All of these vaccines induced a very robust neutralizing antibody response, and they protected animals against pulmonary replication of challenge virus. Many of these vaccines were administered with adjuvants that enhanced the immune responses, and I've listed all the adjuvants that were used. In mice, there was Th2 immunopathology that was described. So this brings me to the concerns about safety of SARS vaccines that I would really be delighted if the DMI community sort of engage in this question, because it is it's a big concern in the vaccine field. It stems from initially an observation with feline infectious peritonitis virus, and maybe Peter can comment on this as from his veterinary background.
I don't know anything about it.
Okay, so there was accelerated and fulminant disease in seropositive cats exposed to a feline coronavirus. This had been seen after infection, after passive transfer of immune serum, and after vaccination. And the mechanism is believed to be an antibody-dependent enhancement similar to dengue. So that was in the background. That's something everybody knew about and was concerned about, because we do not have any licensed vaccines for human coronaviruses and any licensed vaccines for coronaviruses for use in humans. So what was done after knowing what was known about the feline coronavirus? There was a lot of concern about the safety of SARS or MERS coronavirus vaccines. And in vitro, antibodies can be generated by a recombinant spike protein vaccine and enhance viral entry in vitro. And so this was a recombinant spike protein that I collaborated with people in Hong Kong on, that induced serum IgG that blocked the binding of the spike protein to ACE2 and neutralized virus in vitro. But when they tested it on human B cell lines in an Fc gamma R2 dependent and ACE2 independent fashion, there was enhanced entry of virus into human B cell lines. However, the same vaccine, when tested in vaccinated animals, showed no evidence of enhanced lung pathology, and viral load was greatly reduced in the lungs following challenge with SARS, and that was published. The additional concerns are that although we have seen no evidence of enhanced viral replication in mice or hamsters or even monkeys, in the presence of subneutralizing levels of serum antibody following infection, following vaccination, or passive transfer of monoclonals. Despite all of this, the fact that there's no evidence of enhanced viral replication, eosinophils were found in the lungs of mice vaccinated with a whole virus vaccine following challenge. Vectored vaccines expressing the N protein, and this was seen in old but not in young mice. And follow-up studies that were done in BALB/C and B6 mice with inactivated and S protein vaccines with and without alum showed these eosinophils. Notably, this has not been seen following live virus infection, either with the wild type or an attenuated vaccine that we worked on with, that Luis and Manis from Spain has developed. Now, these findings are described as being characteristic of the Th2 immunopathology pattern. And in one paper that was not published, the data were not published or have been presented at meetings, and one paper that was published that I've cited there. Ferrets and monkeys vaccinated with an alum-adjuvanted whole virus vaccine were protected from challenge virus replication, but they did not inflammation in the lungs. So they didn't see eosinophils, but they saw enhanced interstitial inflammation. So this raises the question about the safety of a coronavirus vaccine in humans. If we vaccinate people with a coronavirus vaccine, will subsequent exposure to the same or a different coronavirus result in worse disease? So what's being done about these concerns? The WHO has formed working groups to consider this problem. CEPI has engaged, and I should say that there are only 2 SARS vaccines that made it into clinical trials. They were small phase 1 clinical trials. One was an inactivated vaccine that was tested in China, and the other was a DNA vaccine that was made by the NIH VRC that was tested in phase 1. The NIH extramural NIAID had planned a much larger clinical trial of an inactivated vaccine, but by the time they were aware of all of these findings in mice, monkeys, and ferrets, and SARS had gone away, the risk-benefit calculation was such that they made a decision not to proceed with the clinical trial. We're in a very different place today. The risk-benefit calculation is very different. And so CEPI has actually engaged an organization called the Brighton Collaboration that works on vaccine safety to review the animal data and to make recommendations on how to evaluate the SARS-CoV-2 vaccines for these concerns. Which animals should be used, animal models should be used? Should phase 1 clinical trials be delayed until studies are completed in non-human primates? And what are the considerations for phase 1 clinical trials? So I have been sitting in on these discussions with CEPI and the Brighton Collaboration, both on the animal model side as well as a clinician looking at developing the case definitions so that everybody's on the same page when these vaccines are taken into phase 1 clinical trials. And so some of those the manuscripts should come up very shortly. For immunologists, I think there are some really big questions. What is the mechanism underlying these observations? What is the nature of the observed inflammatory changes? And what is their significance for humans? With that, I will close. And all of this work was done more than 10 years ago, like 15 years ago. So it's been a trip down memory lane with a lot of assistance and great collaborators.
So Canto, you said that the inactivated vaccine, we're getting some Th2 type pathology, not with the recombinant protein?
They saw it actually in the end with recombinant protein as well.
Okay.
And with anything, so the first studies were inactivated whole virus.
Yes.
And a vectored vaccine that had the nuclear protein but not the spike. Spike alone was fine, the N protein was not. But then there were subsequent studies in which they've seen this, and one of the concerns that Barney Graham and others have raised is that essentially if anyone who wants to can actually show some Th2 disease in a mouse, and they said to be very, very careful that when you challenge the mouse, that say for instance, if the vaccine was made in Vero cells, or even the excipients in the tissue culture fluid and everything else could be the source or the cause of that immunopathology.
So the spike that was safe
gave
you neutralizing antibody.
Yes, all of these strategies gave us neutralized everything except N.
How severe was the Th2 type pathology?
Not very.
Well, yeah.
And there are a load of publications. I'm happy to send
Because
them out
we
to
often-
you and you can send them out.
We often beat, I mean, we're immunologists, we often beat up stuff, right?
Yes, and that is one of the concerns of CEPI and the Brighton collaboration. They want to make sure that these concerns don't halt the process towards human clinical trials. And I suspect, and don't quote me on this yet, I suspect that phase 1 clinical trials will happen in parallel with evaluation of the safety.
I'm very much of that mind that phase 1 clinical trials should happen in parallel, even if there is some risk, because this is such a
That's right, but
high risk situation.
It has to be with informed consent in
Yes.
people that are very healthy with, carefully
My hand is
selected.
up for the first trial.
Thank you. Thank you very much, Kanto. That's a fantastic way to set the scene for the next few presentations, and really appreciate you giving us a talk here. So I'm sure there's 64 people playing in their own offices at the moment. So the next speaker we have is Catherine, who's going to tell us about some work examining what's happening with the immune response. What's happening with the immune response in patients who have been infected with COVID-19. So thanks very much, Catherine. It's over to you.
Thank you, Dale, and hello, everyone. I'd like to thank also Dale and Sammy for inviting me here today to share with you some of our recently published and some of unpublished data.
Oh, that's me.
Oh, that's you doing that?
Yeah, I don't know why it stopped.
Mm.
You can just give me a nod and I'll.
Okay.
Yeah.
Excellent. Thank you. So, as many of you know, my lab's been interested for more than a decade now in understanding immunity to newly emerging viruses, and especially influenza viruses. And that started in 2009 when we had an outbreak of the swine flu pandemic. We've started looking at immune responses, human immune responses to the new pandemic virus, and mainly at T cells at the time. And we were the first group to show epitope-specific CD T cells can respond, previously pre-existing memory cells can respond to the new pandemic virus, and there was some cross-reactivity between the 2009 and 1918 influenza viruses. Then when in 2013, avian H7N9 influenza emerged in China with mortality rate of more than 30%, we've done some really exciting work in collaboration with Fudan University and Jianting Xu to answer two main questions. One, why some people, what immune responses drive recovery from severe influenza infection? And the question number two was why some of the patients die of severe influenza disease. Okay. So then, because of all of those, our interests, in 2019, when Kem Siemens and Irani Rajan invited me to be a part of the CETREP, I was very excited and joined them to lead the immunology part of CETREP. And CETREP is led by Irani, who's done an amazing job at getting the platform off the ground, setting up all the protocols and ethics so they're in place. So the platform is really an active framework. with the potential to respond to future epidemics and pandemics like COVID-19. And what it does, enables a broad range of biological sampling to be done in return travelers with new and unexpected infectious diseases, which is what happened with COVID-19. And as you can see here, there are a number of experts. There is a broad range of expertise from both the Doherty Institute and RMH within SARS, and we've been driving the immunology part of it, and it was myself and one who really helped Irani to establish the protocols for immunology and help with the ethics for isolating PBMCs. Difficulties, yeah. So, and that's the people involved in SARS from Doherty Institute and across different hospitals as well as the funders. So when so then when at the end of January, when we started having first cases in Australia of COVID-19, Irani has recruited the first COVID-19 patient, and it was one of the first Australia's patients. It was a 47-year-old lady from Wuhan, Hubei province in China, that presented to the hospital with a 4-day history of lethargy, sore throat, cough, chest pain, and fevers. She lived in Wuhan, but had no contact with the city market or any cases. The patient was otherwise a healthy female, non-smoker, and not on any medical medications. And that's what we recently published as the case report on this patient. Yeah, I can't use the keyboard. Oh, N.
Yeah.
Okay, I'll use N from now on. And for immunology, what we get for immunology, 6 mils of blood at different time points. And here from these patients, we got blood from day 7, 8, 9, and 20. The majority of blood goes for biobanking, PBMCs and plasma, and it's available to a broad range of researchers. There is a form, and for the research proposal, which needs to be approved by the SARS executive. We were granted access to 400 microliters of blood, so that's for the 2 whole blood stains, and to do the immune analysis. And these are people from my team that really contributed to this work. One, I told you, she drove SARS from the immunology side since 2018. Marius, who actually designed the panels so we can utilize those 400 microliters of blood as efficiently as we could, and get as many answers as we could from such a small volume of blood. And Caroline set up my lab for the for working with COVID-19 patient blood.
Sorry, Catherine. Apparently there's more noise again. But everyone listening, please make sure their microphone is muted. Apparently there is some noise. Thank you.
Okay, so the first thing we looked were, so it was the immune parameters we looked at were based on our previous knowledge from influenza. And the first 2 things we looked at were antibody secreting cells, obviously rapidly producing antibodies, and follicular T cells, PD-1 ICOS expressing. And here, as you can see, when we get the first blood sample on day 7 after disease onset, we started seeing both antibody secreting cells and follicular T cells, which rapidly increased by day 8 and then 9. So the peak of antibody secreting cells was by day 8, and follicular T cells were still going up to day 20 when the patient had full recovery. Comparing that to our previous data from influenza, you can see here we're looking at quite a few patients. What we see is that similar to these COVID-19 patients, the peak of antibody secreting cells is actually around day 8-9, with follicular cells happening around the same time, but lasting for longer. Such an increase in antibody secreting cells was associated with progressive increases in plasma IgM and IgG antibodies in these COVID-19 patients, as shown very nicely by Julian and Suellen from Vigil by the immunofluorescence antibody staining for IgM and IgG bound to the SARS-CoV-2 infected viral cells using the plasma cells at 1:20 dilution. So apart from looking at antibody secreting cells and follicular T cells, we obviously were interested in seeing in T cells, both CD8 and CD4 T cells. This is a new virus. We don't have tetramers to assess the specificity of those T cells. So we did staining for CD38 and HLA-DR because the co-expression of both markers is to date the best phenotype of activated CD4 and CD8 T cells during a viral infection, and that includes influenza, dengue, HIV, and Ebola. And as in other viral infection, as you can see from day 8, there were really beautiful, robust responses, populations of both CD8 and CD4 T cells. And looking at the granzymes and peripherin characteristics of activated T cells, we HLA CD38 T cells, which you can see here in color. We can see that there's a substantial proportion of those activated CD8 T cells, especially CD8 T cells that emerged on day 8 after the disease onset, had those killing molecules. And the data were quite similar to what we've seen previously in the H7 and 9 infection. As we've seen before, that in patients that recovered from H7 and 9 had large populations of CD38 HLA-DR expressing CD8 T cells, which then went down when the patient recovered, which happened in this COVID-19 patient. However, in cases in patients that succumb to fatal H7 and 9 influenza, as you can see, those CD38 HLA-DR expressing CD8 T cells were not transient. There was a prolonged expression in those patients. In some patients, just before they died, even 50% of the cells had the expression of those markers. So we really think that the expression of CD38 HLA-DR can distinguish, the kinetics can distinguish between different disease outcomes. Okay, so in this COVID patient, we also look at monocytes and K cells, and we found no markedly increased increases in either of those cell populations. We also looked at this cytokine chemokine profiles and found no dysregulation of cytokine chemokine milieu at day 7, 8, and 9 of the infection. And for comparison, you can see here healthy an individual infected with the seasonal coronavirus or flu-infected patients at the same time points. So it seems like there was no dysregulation of cytokines in this patient. And we've looked at those, the cytokines that we've looked was as we've done quite a bit of work in influenza patients across different ranges of infection. And what we find is that cytokines, chemokines can be quite a good predictor of disease severity, both at the composition and at the composition level and the level of amount of those cytokines, as you can see here, across different disease severity level. So the last thing that we've assessed in this COVID-19 patient was the IFTM3 genotype, and that's because IFTM3 is probably one of the key markers for disease severity in influenza, and there have been several reports on this. And there is a SNP 1 to 2, and it leads to a truncation of IFTM3 by 21 amino acids, and making the IFTM3 less functional. And those people in influenza seem to have more severe disease. And this patient had the CC genotype. And obviously, this is just one patient, but we thought it was worth reporting so people can possibly look in larger cohorts. So there was the case report. So in this patient with a mild to moderate disease, we obviously observed robust immune responses on 3 days before the patient's recovery, and there was mainly antibody-secreting cells, follicular T cells, and activated CD8 and CD4 T cells. Since that report, we've looked at the number of other patients, and I'm just quickly going now to show you 2 more cases. These patients through our cohort, TC cohort from the Alfred Hospital, that we've established as an HMRC program grant, and it's a wonderful collaboration with Professor Alan Cheng and Simbos. And from the DC cohort, we are recruiting patients presenting to the hospital with influenza-like symptom illness and respiratory infections. And what we are capturing that around 2/3 of those patients are positive across different influenza subtypes, but we also capturing other respiratory diseases. So we've had 2 additional COVID patients with mild to moderate disease. One was a 60-year-old patient, and we had bloods at day 4 and day 6. At day 4 after disease onset, and the second patient was a 25-year-old female. We had bloods at day 2 and 6 after disease onset, and both patients were discharged at day 6. And pretty much very similarly to what we've seen in our case report, we see, especially in the patient that was 63-year-old, amazing antibody-secreting cells, activated follicular T cells, CD8, CD4s, and that's at day 4 after disease onset. Patient number 7, the second patient, the 25-year-old patient, the populations are smaller, but the patient recovered as well at day 6 after disease onset. And the same, that didn't come out well, but yeah, we found that the activated CD38 HLA-DR CD8 T cells had really nice granzymes in periphery, less in CD4. So, in conclusions, what we found was that although COVID-19 is caused by a new virus, we found robust immune responses in blood of COVID-19 patients, and it is prior to patients' recovery. And these are 4 populations: antibody secreting cells, follicular T cells, CD38 positive HLA-T, CD8, and CD4 T cells. Those T cells, the way they emerge in the blood, are really similar to what we've seen over the last 6 years with influenza patients. It is not to say that the overall immune response is the same, because all of us had the prior immunity to influenza, including antibodies and so on, and cross-reactive T cells that we got after previous influenza infection or vaccination. But the way those populations emerge in the blood are very similar to influenza. We found IgG, IgM antibodies progressively increased in plasma. Again, we still do not know what's the nature of those antibodies. The CD38 HLA-T cells had high levels of granzymes, peripherin. The patient case, we found no cytokine dysregulation in this mild to moderate patient. The case report really, what it suggests to us, the patient had really a textbook immune responses that were rapid and really controlled the inflammation, and patient expressed the IgG3. So what our data so far provide it's an important step towards understanding what drives recovery from mild to severe disease. Obviously, analysis in larger COVID-19 cohorts are needed. But now, I think what we have are the basis for understanding what's lacking, impaired or overactive in patients with severe and fatal COVID-19. And the most important question, obviously, to us as immunologists is whether we can establish, we know that the immune response is driving recovery, but can we establish immunological memory for subsequent infections? So I think I've acknowledged everyone on the way. But again, wonderful people from my team doing the work. We've worked with everyone at the Doherty Institute, Iranis, Steve and Ben as clinicians. Wonderful collaboration with Vigil, Julian, Leon, Sulan, and Mike. Sharon, a great supporter of SETREP, our immunological study, and provide expertise on the case report, separate SETREP members, and our collaboration with Alan Cheng and continues. Thank you for listening.
Thank you very much, Catherine. Fantastic presentation, and really important to start to get a bit of a sense of how the immune system is responding in these patients. Really critical for understanding how to make the best vaccines. In case people missed the start, so we're planning to have questions at the end after all the speakers have talked. So there'll be a panel discussion involving all speakers in up to 2 more talks. So thanks very much again, Catherine. Damien, so the next speaker is Professor Damien Purcell. And Damien will tell us about what's currently going on in vaccine development, both here within Australia and internationally, I think. Yes. And can we mute? Can we do stop others from turning their mute off? Yeah. That will just make sure.
Okay. So
Do the participants.
Oh, okay.
I think new people come along and they have the microphone. So you have to unclick that box to allow participants on. Yeah.
Okay. So this is a very busy space for activity in the last 2 months from a standing start with a new virus. So as Dale said, I'm going to talk about vaccines against the virus, SARS-CoV-2, and prevent the disease COVID-19. So often people talk about the COVID-19 virus. So as a virologist, we'd like to define our target, and I'll be talking more about that. But there's one thing that is an issue with vaccine development, and as I'll mention at the end, we're doing this in an environment where there's a lot of activity generating, testing and developing antiviral drugs, immunotherapeutics, passive vaccination with monoclonal antibodies, some of which Hunter touched on. So, when we come down to looking at prophylactic vaccination, I mean, this is something that's actually probably going to be the slowest to develop of all of these other strategies. So, as we move forward with developing a prophylactic vaccine, we might be testing this in the context of overlaying clinical trials that are currently looking at antiviral drugs and immunotherapeutics, passive vaccination as well. So forthcoming difficulties with how we will move forward with the evaluation of any vaccines candidate coming through. So what is the target? So coronaviruses are really not extensively researched in Australia. I think at ARL there was one guy working defining coronaviruses after SARS came along. Jason McKenzie's got a publication on SARS coronavirus electron microscopy. But apart from that, there's very few people studying this virus, and the reason is actually a very complex virus. It's a positive sense RNA virus, RNA genome that is about the biggest RNA genome that we have of all of the RNA viruses, around 30 kilobases in length. So the virus has 3 proteins in the lipid bilayer: the spike protein, an envelope protein which acts as a viroporin, and there's a matrix protein, membrane protein, that assists in actually helping this virus bud into vesicles and egresses from infected cells by exocytosis within vesicles, so a bit like flaviviruses. So the genome is huge, as I said, and it makes a lot of very good immune targets if you're looking for epitopes for T-cell vaccine with this massive RNA-dependent RNA polymerase. But the main target is the spike protein because, as we've heard from Catherine and from Hunter, that antibodies against the spike protein look like they're actually quite effective neutralizing the infectivity of the virus. In addition to the spike protein and the other structural proteins, there's a series of other genes that code for different proteins that probably have immunomodulatory activities. So the spike protein is probably the best defined target. It's a trimer that exists on the surface of the virus particle embedded within the lipid bilayer. And already we have a structure of the spike trimer published in Science a couple of weeks ago, a remarkable fast effort by the research team. And the structure was conducted on a stabilized spike trimer. So a lot of lessons have been learnt about how to begin producing subunit vaccines. A region of the spike itself that is the dominant interest is the receptor binding domain, the RBD, that interacts with the cell receptor, the ACE2 protein, so angiotensin converting enzyme 2, which has pretty limited tissue distribution, upper airways, very high expression in the gut, in kidney, a few other organs, central nervous system, and male reproductive tract predominantly. So the virus, or different viral targets for vaccines bring their own complexities for vaccine manufacturers. So things that we've been focusing on a lot of research over recent years, HIV and hepatitis C, use the strategy of rapid evolution. So this is a concept that RNA viruses have that assists in their escape from particularly antibody-mediated responses. So there's been a lot of effort recently to map the evolution of the genetic sequences of this virus, and sort of quite pleasingly, even in the first publications, paper out of Lancet, looking at the point changes across the genome, very, very infrequent. And in the all-important regions, such as the receptor binding domain, this is looking across more than 300 genome sequence, you can see that there is really a very low level of change between the various isolates. So it's comparing SARS-1 and SARS-2 on the top line, but below that, this is really the sum total of the amino acid changes across many, many sequences.
Where's the receptor binding domain in that?
In the
sequence, yeah, is that changing or is that stable in these different variants?
That is, well, I don't know about the function. I'm not sure whether that's investigated. That's just coded sequence from sequenced isolates. And this is the receptor binding domain. Sorry, where is it here? Yeah, so probably not the ideal figure, but that's sort of summarising a lot of sequence. So Lee Hardy in my lab has been monitoring that and has produced that. So it's probably not publication quality, but just so. So low number of changes in particularly spike protein, where we anticipate that being the dominant target for antibodies. And the reason for that is that the virus has got this error-correcting enzyme activity. So the RNA-dependent RNA polymerase targets has an error-correcting activity, unlike retroviruses and hepatitis C. So it's quite fatal. Now, one of the things that might be interesting is some of the antiviral drugs that target the RNA-dependent RNA polymerase, the nucleosides. So some of them have the activity of mucking up that error correction mechanism. So remdesivir is one that you might be reading about, and it might actually drive additional mutations. And there's something to bear in mind as we're studying the virus. As these antiviral drugs roll out, perhaps they might actually drive additional mutation of the virus and might confound some vaccine investigations. All viruses have countermeasures against the immune response, and this virus is no exception. It has quite a number of non-structural proteins, and the activities of those require further investigation. But one thing that's sort of quite interesting is at the 3' end of the coding sequence, this virus makes these small viral RNAs. They're non-coding RNAs that seem to have the activity of targeting messenger RNA target cell, and have been associated with driving the inflammatory process in lung paper. So this might be tied up in the ADE or the inflammatory response to eosinophilic infiltration into lung, perhaps driven by the virus itself. So not necessarily a viral RNA, it's not necessarily an immune correlate per se. So you always have to bear in mind that the virus has got its own contributions to how the immune response will develop. So if we look at the concepts that are out there, and this is pretty much a data-free zone at the moment. There's a lot of activity. The lead candidates in the world at the moment have been mostly funded by the Coalition for Epidemic Preparedness Initiative, CEPI, which has backed a couple of platforms that can be rapidly adapted across to any new virus. So fortunately, SARS and MERS were on their list. So quite a few platforms have already investigated coronaviruses as a meaningful target. So on the scoreboard of things that are underdeveloped, some of these are quite well developed, others poorly developed, and I'll just go through what I know about each of these things. So fixed or killed vaccines, these are pretty much not favoured for the exact reason that Kanta presented before, the feeling that these elicit the ADE response, they drive pathogenesis, and there are unfavourable immune activities encoded within these vaccines. Maybe they're viral RNA mediated, we don't really know. I think that the jury's still out. Maybe they're also antibody mediated, and I think those FC mediated uptake into monocytes and carriage of virus into other tissues, such as central nervous system, is something that is a consideration that might be listed for these kind of vaccines. And again, not great data that I know about. Live attenuated vaccines is something that a few people are talking about. So there are similar coronaviruses that have been developed as veterinary vaccines, and one that's getting a bit of airtime at the moment out of Israel. The Doherty Institute there, they've developed a chicken coronavirus vaccine, and they feel that they can substitute into that the spike protein coding sequence because it has the same, uses an ACE2 receptor, and they feel they can engineer that into a live attenuated vector for the human virus. But I think, there's a lot of work to do. They're beating it up quite big, and you see quite a bit of press about it, but not a lot of data, and it looks like it's got a long way to go from my perspective. Viral vectored vaccines. Now, this is an area that's making quite a big push forward. In the CEPI stable of vaccines, there's adenovirus vectored strategy. So this is a group based in Oxford. They're partners in our European AIDS Vaccine Consortium, and they're using the chimp adenovector. So in the HIV setting, this has very impressive T-cell responses, and also is very, very good at assisting potent neutralizing antibody responses. So I haven't seen the data, but rumour has it that particularly the chimp adeno vectored vaccine is moving forward fairly quickly. The advantages there, adenovirus has got a very good safety profile. Many clinical trials in the setting of HIV and other complex experimental vaccine systems, those systems have demonstrated good safety, which is clearly a paramount consideration. So I expect that we might see animal studies with the adenovirus vectored vaccines coming off fairly soon. I think that's the strategy that these viral vectored vaccines would need to go through. So initiating studies in animals first, because of the potential risk when you mix bits of viruses together that you might actually generate sort of unknown and untoward responses that really do require a decent animal model to test before moving forward into humans. Now, virus-like particle vaccines, this is something that we're focused on intensively here at the Doherty with our Jack Ma Foundation. So I can't elaborate extensively on the technology publicly, but certainly published work from Joe Teresi and also from other labs, Jason McKenzie's group, and also from the Dave Jackson, Brendan Chua group, have demonstrated that virus-like particles can be produced in flavivirus models using an adenoviral expression system. So it's sort of like a Russian doll. The adenovirus, which can be produced in massive quantities, is capable of initiating the expression of virus-like particles in target cells. So the advantage of this system is high-level production, and it's quite amenable to genetic manipulation. So I think the concept is to express the SARS-CoV-2 spike protein, maybe in combination with envelope, maybe also including the matrix protein, to assist in the egress of those virus-like particles into the endosome pathway for exocytosis from cells. So vesicles loaded up with virus-like particles that will be shed from transduced cells within the patient or the test animal. Other virus-like particle platforms, so in my lab we've generated an HIV or lentiviral-based virus-like particle that we can load up with different envelopes. So we've obviously been focusing on stabilized HIV trimers, but this is also a system that would enable packaging of spike protein and engineered in a way so that those particles egress through a different pathway from the plasma membrane and budding out from cells. So it's a similar concept, making a virus-like particle, this one structured around the HIV gag proteins, but departing the cell by budding off from the plasma membrane instead of being exocytosed from vesicles. So these are obviously newly funded and just coming from a standing start. We're hoping to push these forward. And these offer some benefits in potentially, as a particle, have perhaps different methods of stimulating immunity than the next thing I'll talk about, which is recombinant protein vaccine. So recombinant proteins are very attractive. We have human vaccines made from recombinant proteins that are successful, particularly the papillomavirus and the hepatitis B vaccine, although they also sort of fall into the virus-like particle vaccine category. But the recombinant protein vaccine that's getting the greatest run is one that was adopted from technology developed in the University of Queensland, the so-called molecular clamp. So this is Paul Young's group and Keith Chappell and Dan Watterson, who's will be here soon to talk about this approach. But the concept is to take a common component, a trimeric protein derived from another virus, stick it on the base of monomers of various different viruses, and here MERS is one that was made and been tested, and then use this to stabilize proteins into a conformation that accurately resembles the structure of the functional trimer protein that appears at the surface of cells. So this is like a spring-loaded machine that engages the receptor, and if this pops open, as we've found in many studies with HIV, we've looked at this for a long time. The best vaccine responses we've seen in HIV, and similarly with HTLV, if you don't stabilize the trimer and you allow it to open up, you get very poor responses. You get non-neutralizing antibodies mostly, and you get a lot of antibodies driving cell-mediated immune functions like ADCC or ADCP, but you can get very good neutralizing antibodies from these stabilized trimers. So recombinant protein vaccines, probably this will be the first one coming through in Australia. It had a flying start thanks to very good funding from the CEPI funding. So they've made candidate. They had a lot of different structures that they were working with, and that produces efficiently. They've got a method of purifying it through that the clamp with a monoclonal antibody to bind that region, so they can produce it at high levels. And it's moving forward and might be in a race with the viral vectored vaccines from adenovirus to see which one is the first of the CEPI candidates, protein-based candidates, to make it into animal efficacy studies. The remaining platforms are built around nucleic acid. So CEPI have funded a nucleic acid platform from Inovio. The thing with their system is that they have devised a method of very efficiently delivering DNA, so it's taken up in cells and expressed at reasonable levels. So that's not a trivial exercise. Many DNA vaccines work brilliantly in mice, but as you step into another animal system, those DNA nucleic acid constructs that express the relevant proteins have failed. So Inovio are hoping that they can overcome that, and they're moving forward with this concept with CEPI funding. Now the last one that is out there, so 2 groups funded by CEPI, one with a company called Moderna, thank you. They've got press recently with a mRNA essentially that expresses viral components. Now I haven't been able to determine exactly what that expresses, whether it's large slabs of the virus or just the spike protein, but that's moved forward into the arms of people because there's very little novel cytotoxicity or toxicology associated with a messenger RNA. They've somehow got approval to move directly into people without extensive toxicology studies on the product. Another one we know about, also funded by CEPI and also in the European AIDS Vaccine Initiative Consortium. In fact, the head of that, Robin Shattuck, based out of Imperial College London, he was here at the end of last year giving a seminar in the Doherty. He has an advanced mRNA platform expressing spike protein and I think making stabilised spike protein, sort of looking a little bit like the one I was showing you with the plant trimer, but in their case they've applied all of the learnings from HIV engineering to produce a stabilised spike that will be the best candidate to elicit neutralising antibodies according to the results obtained with other viruses. So making the candidate is one step, and some of those things have moved very quickly, but the bottleneck is really all the subsequent things that have to happen. So any vaccine that's moving forward has to demonstrate a very robust level of safety. So we've already heard from Cantor about the antibody-dependent enhancement. We don't fully understand all of the signals that are associated with the immune responses that signal that event, but that's something that we've applied in our MRFF application with Amy Chung and Catherine to sort of tease apart what defines a protective antibody response. So something like virus neutralization is the most obvious thing, but negative signals, perhaps an FC-mediated uptake by monocytes and carriage into other tissues, other organs might be a negative safety signal that we need to avoid with our vaccine. T-cell responses, obviously Catherine's demonstrating meaningful responses and whether we can map those better, understand what the neutralization epitopes are. We can map those on mutant strains of the spike. We can present all the different variants of spike that come along so we can know the breadth of protection that we might elicit with our vaccine candidates. And then finally, the steps of animal efficacy studies. So ARL have taken the Doherty isolate and they've successfully infected ferrets with the Doherty virus. They've elicited some inflammatory responses there. So they feel that that's looking like a suitable animal model, but we know that ferret's obviously not human, and in particular the affinity of the ferret ACE2 receptor for the SARS-CoV-2 spike is much lower affinity than against the human receptor. So it might be much easier to achieve neutralization in the ferret model than it is in human. Toxicology studies are pretty standard. Any vaccine candidate will have to fill the hurdle of toxicology before it gains approval to enter into human clinical trials. So, this is where the rubber meets the road. And as I mentioned at the beginning, we're going to have to think pretty hard about how those trials are designed. It's probably the first time we might have a prophylactic vaccine candidate coming along where we might be able to overlay things such as monoclonal antibodies and also redirecting the response to potentially antiviral therapies if things go off track. For example, inducement of these antibody-dependent enhancement of pathogenesis. So it's likely that there's going to be a lot of clinical trials overlapping, and how they're all going to work is going to take a lot of thought and clever design from clinical trialists, and I think the Doherty is well placed to do that. So that's all from me. Finished ahead of time.
That's wonderful, Damien. Thank you very much. A great overview and drill down on some of the vaccines with all these different approaches. It's hopeful that ultimately there will be a vaccine that will be able to protect us at least a few months from now. But fingers crossed on that. So thanks very much, Damien. Our next speaker is the man himself, Professor Peter Doherty. Do you have any slides, Peter?
No, I'm not showing any slides.
Okay. So Peter will be giving us his thoughts and closing comments on where we stand with this infection. Over to you, Peter.
Okay. Anyway, everyone's hearing me okay?
I can't talk. Can I mute?
I'm just going to give a very brief kind of sum up. Clearly, this is the first time we've done this, and this is an evolving process. So we'll be looking for feedback from you and towards developing a more interactive type format. So this is just an experiment. Okay, we're all experimentalists. Just to say something about the disease itself, this is a disease where 80% of people are going to be fine. They get a fairly mild infection, and in fact, there may be a much bigger background of asymptomatic infection, which nonetheless may still transfer the disease. You must realise that all the testing that has been done so far that's been published is all done by the PCR test. So we're only detecting virus. We're not detecting recovered individuals. We don't have a broad screening serum antibody test. It's not that we don't have one, we have a fluorescent antibody test, but we need a rapid ELISA. We need something like a blood pinprick ELISA or something of that sort. Those tests are under development, both here, and I mean in Melbourne. The Doherty Institute is particularly involved, and of course globally, because this whole thing's going globally. The other thing about this, of course, is we are in a pandemic situation. We have to move fast and we have to move decisively. And we don't want to endanger people, but we have to be a little bit dramatic at times, I suspect. Now, with respect to susceptibility, most people are fine. Most people who are in their 20s, 30s, 40s are going to be fine. Doesn't mean they're all going to be fine. We've had severe disease in those age groups. We've had deaths in those age groups, particularly when people are massively overstressed, and we're worried about our health professionals, because even now, some people are starting to get exhausted, particularly at the lab level, because we've been ramping up so fast in testing regimes. So, and children on the whole don't seem to be very much affected. We don't know much about their role in transmission. Still, there have been some small children with severe disease. So it's not as though they're out of the equation. The people at risk, as most of us understand, if you're in your 70s, you're about 3 to 4 times as much likely to die as if you're younger. And if you're in your 80s, you're 5 to 6 more times more likely to die. I'm in that category. So I think one of the points of mentioning that is when we're talking about a vaccine, we're talking about a vaccine that will need to be given to a large number of people who are likely not to get severe disease. So that's got to be a pretty safe vaccine and so forth. Maybe we need to be thinking about another vaccine that's maybe progress more quickly to give into the high-risk group, where we might even take more risk. And that's a serious discussion, of course. And the other thing is, of course, is that the high-risk group being elderly, we don't necessarily know how well they will respond, because we always have a problem with flu vaccines for the elderly. It's very variable, and our naive responses, of course, are not that good. Now, we have heard, Catherine has told us something about what the disease looks like. Nothing particularly remarkable about it for a disease of that type of severity, but we haven't yet looked in that depth and that detail at severe cases. And so that's going to be very interesting. There's quite a bit of hint of that perhaps cytokine storm is involved. There's some results that are being trialled with anti-IL-6, I think also with anti-TNF. It's very clear that people should avoid ibuprofen. That is clear now, I think. And anti-inflammatories may be part of the disease, but not the non-steroidals. There is also a bit of a phenomenon of early infection, people seeming to get better and then relapsing. And part of that relapse can be associated with acute cardiac death. That may be associated, that's probably electrical, may be associated with the fact, of course, that the heart has a lot of ACE receptor. We don't know. We've only known about this infection for 3 months, and we're already an extraordinary way along the road. So our institute's involved in many levels, as you've heard from Damien, as we've heard from Catherine and from Dale, and what we'll be wanting to know is what other people are doing, what you're interested in doing, how you're interested in coming on board with some of this, and what contribution can be made. We know that other institutions, for instance, are focusing on making monoclonals and various other things. So I hope we can use this as a general awareness forum and to get everyone in the immunology community on board and doing what we can, because I know we all want to contribute to this. So thanks. Thanks to our speakers.
Thank you very much, Peter. Now, this is going to be a tricky exercise. We're going to try and see if we can have questions from the people listening. I think there's a function where you can wave your hand or put your hand up.
There's also a chat.
Or
chat, yeah.
Okay. So please, if anybody has a question.
There we go.
Well, there's already, it's all about little bits of
This is coming.
Okay, so if anyone has any questions, type them into the chat thing, or you can, I think, unmute your microphone. Can they do that now? No, they can't. Can we let them?
Yeah, we can let them.
And then speak. Maybe you can wave your hand first. I think there's a wave your hand function somewhere. Okay, Kim. Speak, please.
Can you hear me now?
Yeah.
Yes.
Thanks very much. Thanks for this very interesting meeting. I have a couple of specific, probably rather simple questions, maybe things that I missed along the way. But firstly, as far as the antibody-dependent enhancement story is concerned, it was unclear to me the extent to which this was associated with particular types of vaccine. It seemed like it was originally described with a killed virus type of vaccine, and then, but also with the spike candidate, which seems to be the best of the protein candidates. There was a story about, in Cantor's presentation, I recall, about this being associated with more rapid entry of virus into the cell. So it does, the reason I'm asking this in a way is because this morning on a similar meeting like this, there was a bit of talk about the Moderna vaccine, and people said, oh, that one could be a dangerous one. And I didn't really quite know what that was about, because I had understood, as you've said, that this is a mRNA vaccine, and I wasn't sure that that was one of the things that was considered dangerous. So,
Thanks, so do we
that's
have
my question now.
A response to that?
Yeah, so I don't think that we should focus entirely on the antibody-mediated enhancement because that was seen in vitro, but none of the vaccines that elicited neutralizing antibody and none of the even when we challenged animals 18 months out when their antibody titers were declining, or with monoclonal antibodies given at serial dilutions, we never found a correlate in vivo. So the data that I showed you from the CAM paper was an in vitro observation that was not substantiated with any enhanced disease in vivo. However, the eosinophils and the Th2 and the inflammatory changes in monkey lungs and in ferret lungs, unfortunately, that paper wasn't published, but those are the ones that people are very concerned about. And they first were described with an activated vaccine and with an nucleoprotein containing vector vaccine on an alpha virus background. Subsequently, Kent Seng's group in UTMB looked at a series of different vaccine candidates, including spike protein, and did see some of these changes. But mouse vaccinologists have told me that we should not halt anything based on those findings in mice. The findings in primates are more worrying.
Yeah. Well, one thing we learned from the HIV world is mice lie, monkeys don't always tell the truth. So we should keep that in mind. Also, this is a relative risk scenario, and that will be very much in mind, I think. If you think, for instance, about cancer therapy with the monoclonals, the immune checkpoint inhibitors, they often switch on severe autoimmune disease. That's something that clinicians can handle. So there's that sort of undertone too. It's just like, especially in giving a vaccine to large numbers of people who would not be seriously affected, we must be very careful there, I think.
Just to wrap up on the ADE, one of the greatest proponents of worry about that is Ab Osterhaus, and he has got a paper that I think is coming out very soon in primates, where he is very happy to say that this activity was not measured in his study. So to have him satisfied would be actually very...
Well, Ab is a terrific scientist, and he's been in this from the beginning. Not so much an immunologist, more a virologist, but we won't hold that against him.
No, we'll hand it back to you.
Okay, we have a few questions coming up on the chat now. So we have one from, I can't tell who it's from, but is anything known about intrauterine transmission? That's from Celine Gupsa. And are there concerns about fetal infections in early pregnancy? Does anyone know?
The only data that I've seen about pregnancy, and I think it's probably not necessarily about early pregnancy, it's later pregnancy. There's no evidence that I've seen that pregnant women are at an unusually high risk. I think it's far too soon to know what the consequences of infection in early pregnancy are. I think we'll have to wait months to see that.
Yeah, I think we got a very clear signal even from the 2009 H1N1 flu that heavily pregnant women were seriously at risk.
Yes.
But I don't think we've seen anything like that as far as I'm aware. I haven't seen it mentioned anywhere.
No.
I did see one report, but I don't remember whether it was in a newspaper or in something more serious of a baby being born who was PCR positive.
In the large series from China, pregnant women were not counted as a particularly high risk for severe disease or death.
Even in early pregnancy, those wouldn't have delivered their babies yet. So.
That's what I mean. You'll be waiting months to see that.
Thank you. So we have a question from Fabienne Mackay. Can the serum of patients with fatal or resolved disease be tested in the viral epithelial assay for BN antibodies?
So we have a microneutralization assay that detects neutralizing activity, and we can test for neutralizing activity. Now, whether we can call it broadly neutralizing is something that, I don't know whether Fabienne used broadly neutralizing intentionally or just as neutralizing. In terms of finding antibodies that are broadly neutralizing across other coronaviruses, I don't know. We have some antibodies that were made against SARS that are being shipped to me to be tested. So some might cross-react, but maybe not. Very often the neutralizing antibodies may be more strain-specific, although it's possible that there's something in the S2.
Data that I've seen sort of indicates that the neutralizing antibodies are pretty specific for the strain. So.
There were there was some sort of rumor or something that the Chinese were using convalescent serum. Do we know anything about that?
I haven't seen any reports.
There has been some hodgepodge
Anecdotal.
tests with convalescent serum. I don't think any clear answers as
Nothing
yet.
systematic.
Yeah, usually not welcome.
One of the points about it, the receptor binding domain is one of the things that varies a lot between the viruses, and so cross-reactive antibodies may not be able to neutralise, because that would probably be the key part you want to target. There was a question from Alex. How similar is SARS-CoV-2 to SARS, and what is known about...
Sorry, Dale, I think that's just been answered by the discussion in the room.
Gotcha, Alex. Thank you.
The same question.
Okay, Kim Mulholland, is the therapeutic use of hyperimmune globulin under consideration? That's a bit also like the question we've just had. So everyone's thinking along the same lines.
I think we had some discussion of this. And I've just heard the secondhand that CSL is looking at that. I mean, at actually separating convalescent serum, but wouldn't be in the standard hyperimmune globulin. But that's a product they provide all the time, so it's right in their area of competence. And one also wonders about adding monoclonal antibodies into that mix.
So I think in some ways a cocktail of human monoclonal antibodies could achieve the same thing.
Yes.
The hyperimmune globulin strategy was tested for influenza at the NIH and did not show clinical benefit.
Okay.
Was that as a prophylactic or as a therapeutic?
That was as therapeutic.
Yeah, I'm thinking particularly of those people in the community who are on hyperimmune globulin normally because they've got defective immune systems. That would be some children, for instance, with X-linked immunodeficiency disease. And it would also be a lot of elderly people with immunodeficiency related to age. So I wonder whether convalescent serum might not be added, or monoclonal antibody cocktail might be added prophylactically to the hyperimmune globulin.
I think there's a lot of interest in making cocktails of human monoclonals.
Yes.
Can I ask about the role of ACE? A lot of people take ACE inhibitors. They're usually elderly. Is there any sort of link?
There's a lot of conversation about that,
Yes.
and a lot of interest in looking at that in severe illness.
The general advice that's been out there is that people who are taking ACE inhibitors should not stop taking them. That's basically the advice. But
there's a theoretical concern that people that are taking ACE inhibitors, that the ACE2 gets upregulated.
yes.
And so there's a lot of interest in looking at that.
So that's why the doctors have been putting it out there, because they don't want people to stop taking their hard drugs.
ACE inhibitors act on ACE1, but ACE2 upregulates in response. So yeah, it could be a negative thing to do. There's a question from Jose Villadangos. What is chloroquine thought to do in this context?
It messes up the acidification of vesicles, so it impedes the import of infectious virus and egress of infectious particles. So, yeah, no in or out of infectious virus.
So I didn't talk about what my lab here is doing, but we're looking at already existing drugs in an antiviral assay, and so we're looking at chloroquine and hydroxychloroquine.
But the medical people are certainly trying to reserve chloroquine as a therapeutic. That's they don't want people to rush out and try and denude the pharmacies of chloroquine, as I understand
You
it.
can't buy it here. You can only get hydroxy.
Yeah, the hydroxy also works, doesn't it?
Better safety.
Yeah.
A lot of tonic water, perhaps.
Yeah.
A lot of gin, yeah.
Gin.
Clear that there are multiple strains of SARS-CoV-2 going around, and that was
I think it's a low level of genetic drift compared to things we're used to looking at, influenza, HIV. I mean, there are some mutations, but it has an error-correcting enzyme that actually makes it a pretty stable
What
target.
the coronaviruses do, and that happened with SARS, was that they can actually have deletions elsewhere in their genome. So ORF8A was one of the things that happened that deleted, and was a difference between the early strains of SARS and later strains of SARS. So those were discrete introductions, reintroductions from animal species. I think what we're looking at in the SARS coronavirus 2 is a bit different. We're we're not looking at multiple introductions from animals, we're looking at a lot of transmission in humans. So it's possible that we'll see something slightly different because of passage through millions of humans. But historically, you wouldn't see anywhere near the level of antigenic or genetic drift as you see in flu.
It's found a pretty sweet spot in humans. It's not too pathogenic, so it's not sort of clearing out the host before it has a chance to spread. So, I think it's just landed in a zone for replication and shedding without needing mutation, which is sort of interesting.
And also, it's just as likely as it does mutate that it would go to a less virulent profile rather than a more virulent one, I think. But as I understand it, there are genetic changes you can use to trace lineage, to trace clonality, but they're nothing very dramatic.
Right. Yeah, whether they mean anything, in terms of vaccine, drug,
because I think
remains to be seen.
The suspicion is that the outbreak in the nursing home in Washington State, which is one of the first American ones because their diagnostic capacity was absolutely terrible, but that it actually does go back to the initial case they'd had 6 weeks previously. And through looking at the genetics of that virus, it looked as though it may well be the same lineage. Yeah.
There's a question from Fabienne. What is the status of anti-HIV and anti-Ebola drugs in trials in China? The updates.
So I hear that the data should be coming out very soon. There were 85 clinical trials registered last month, and I heard there are 180 now, which I'm sure they're having trouble enrolling in now at this point.
It's the only problem maybe with those early trials in China that I'm hearing about. It's very haphazard. The trial design is more salvage therapy for patients. So there'll be a signal, but it might not be definitive.
And what happens if, let's say, the city of Wuhan opens up completely now? Can the virus come back? I mean, are we going to see the same thing again and again before it eventually dissipates? Or?
Well, I think there are a couple of layers to your question. One is that we don't actually know what the animal source was. And we really need to be very aware that this is the third animal coronavirus that has infected humans in the last 15 years. And we need to be much more aware of animal coronaviruses infecting humans. So having wild animals in wet markets, bad idea. And we really need to start thinking about how to restrict that. And I think the Chinese are aware of that. So could it come back because the source is still out there? We still don't know what animal it was in those live markets. But we don't need the animal source in this case. But should everything settle down into a bit lower activity, and we go back to business as usual, I think we are looking at potential for more zoonotic infections in the future.
Yeah, and I mean, it looks as though all three have probably come from bats. I mean, that seems very likely. So the camel one, they're not in live animal markets, but you do see pictures. People kissing their camels. So kissing a camel is a bad idea. Don't kiss a pig. Don't kiss a camel.
One question relevant to that I have is the durability of immunity with even like SARS-1. Do we know whether anyone infected with SARS-1 was susceptible?
So we don't know about any of them being infected again, but people have done long-term studies on the antibody, and the antibody lasts quite a very long years. Yeah, so there are a couple of papers now both on SARS and Mars antibodies. Up to 9 years, they still detected antibody.
Neutralizing.
Yes.
We have a couple of comments. One from Dick Struggle. Takeda, who took over Shire, who took over Baxter's blood business, have declared that they are purifying SARS-CoV-2 specific antibodies from convalescent stem. So that's relevant to our earlier conversation. Terry Nolan, a comment only, the Moderna phase 1 commenced this week with mRNA 1273, an mRNA vaccine against the novel coronavirus, encoding for a prefusion stabilized from the stabilized form of spike S protein, which was designed by Moderna in collaboration with NIAID. Thank you for those comments. Jason McKenzie, chloroquine also interferes with furin activity and thereby producing less infectious virus. Thanks, Jason.
Does chloroquine also have some anti-inflammatory?
Hydroxychloroquine, that's why it's used by the rheumatologists.
Jose says I try this question? No, we didn't. Nigel Curtis, we're planning an RCT of BCG vaccines off-targets effects to prevent severe COVID-19 in HCW. What do the
Healthcare
panel
workers.
think of this idea? Too many acronyms.
Healthcare workers.
Healthcare workers, thank you.
I was glad about Terry Nolan's clarification there, because there was a question from Kim about the Moderna vaccine and a comment about the presentation this morning from Rob Grenfell, saying that it had a low safety profile. So I was there and I heard that, and I was unsure about what was actually being expressed. So if it's only the stabilised spike, I don't know that it's actually that innocuous.
And what's the thought of the delay in the clinical disease? Does that take anybody to take 7 days before a disease becomes severe? Where's the
Why
delay
is there a
coming
delay?
from here?
Don't know what I have seen very little information on the kinetics of viral replication. I've seen very few papers describing what the course of infection is in terms of viral load in the respiratory tract. So I think there's a lot that we don't know, whether it's just a
Slow
long,
growing.
yeah, exactly.
Yeah, and the other thing is, is it going from an upper respiratory to a lower respiratory? And I think with any respiratory infection, we always have to bear in mind just how fragile and delicate the lung is, and how easy it is to mess up gas transudation and so forth. So it's a very fragile organ, the lung.
So I'm involved in Nigel Curtis's study, so I can't comment, but the question about using BCG for off-target effects to protect healthcare workers is one that we kind of...
There's a question from Kim in Holland. I presume the molecular clamp system involves S protein. Does S have the same stability issues as RSV F protein?
So yes, it's the same. It's the S protein. The forerunner of all of these vaccines was an RSV vaccine that had the stabilisation through clamp and apparently solved the problem, according to Paul. So, I think the stabilisation on RSV was successful, and that sort of was the forerunner to take that into the spike in this case.
From Phil to everyone, can Catherine summarise the local biobanking strategy and access for researchers for longitudinal blood samples for patients?
So the SATREP samples are processed by my team, and they get biobanked. There is a SATREP executive, and there is a form for research proposal that the executive needs to approve for using the samples. The samples come from one blood tube, so PBMCs are very limited, but there is some amount of plasma, obviously. Hope that helps.
Thank you. Yeah, that's good.
And Irani is in charge, so please contact Irani if you have questions.
Fabienne adds a point. Chloroquine also inhibits TLR9, and this has been shown to have a role in preventing multimicrobial sepsis. So chloroquine has multiple modes of action that may
also messes up autophagy and other membrane trafficking functions of the cells.
Yes. Yeah. So those trials will be very eagerly awaited. Something as simple as chloroquine can help slow this infection.
Probably worth mentioning that in the Doherty that Steve Tong is planning a clinical trial with hydroxychloroquine and the HIV protease inhibitor, factorial, which should be going soon.
So he's asking whether there's a risk for secondary pneumonia, or is there any reports on?
I have not heard, but I cannot answer that specifically. I think we'd have to go back and look at the large case series about how much of a risk of secondary pneumonia there is. Don't know.
Somewhat relevant to, I think, the later stages of disease is what Peter touched on about the cytokine storm. And I think there was a very encouraging results out of an early clinical trial in China, where they took 20 patients who were at a quite severe stage of disease. They were on ventilators, and they took tocilizumab, I think it is, anti-IL-6 receptor. Within a day, most of those patients' fevers dropped down to the normal range, and within 2 weeks, almost all, if not all of them, walked out of hospital. So this concept that at a late stage, the hyperactive immune response, at least in a subset of patients, may actually be contributing to mortality is, I think, really important, because there's a lot of drugs around that can help to inhibit these.
Maybe lack of regulation. I mean, is a Treg response delayed? Because it's very unusual for an innate type of response. You get quick and hard. And here you have this delayed one, and then all of a sudden you get an overshooting response late.
Yeah, it is unusual. And it may be that it's there early and just the virus is down deeper later or something. I don't know. It's an interesting question, and it is intriguing that anti-interleukin 6 or anti-interleukin 1 might be doing something late. There's also a bit of sort of, again, maybe just rumour that maybe the anti-TNF that the rheumatoid arthritis patients are on might be useful too.
Yeah.
But we don't know. I think it's just so early.
I think looking at interleukin 6 levels in severe patients is one thing that people will be starting to do, and also ferritin, which seems to go hand in hand with the IL-6.
I think it's really important that Catherine gets some severe cases to look at, because we'll get a pretty good readout from that analysis about what's happening. So I really do think that's important. So anything that can be done to facilitate Catherine seeing severe patients. Yeah. Because we have a better analytical system than almost anybody else, quite frankly.
I think a question just was lost up. Even higher. So a question from Alex. Nigel or Cantor, is there any data to say that different mortality rates between countries may correlate with BCG vaccination rates?
I think Kim's answered it right after that.
Kim's answered it underneath. It's not routine in Italy, but 100% coverage in China. It's interesting. So I wonder what it's like in some other countries like South Korea. With the disease easing off. From Josh Sawicki, the cytokine storm described for COVID-19 looks a lot like what paediatricians recognise as secondary HLH, well recognised in H5N1. Yes, I think it has been described as secondary HLH in the paper. The paper just came out in, I think it's China Archive, so you can read about that. Kim Mulholland, very few PED cases in Italy that we hear about. So I think we probably need to at least start to wind this up because we have some people who have other appointments that they need to go to. So I really would like to thank everybody who's made the effort to give a talk today. Cantor, Catherine, Damien, Peter, thanks so much for giving your time. Everyone's time is really precious at the moment, and I think this has been very helpful. We will look at having another one of these in a little while to keep people updated. Sammy Badui, who's the co-immunology theme leader, has also just left, and many thanks to Sammy. He's working with me as trying to coordinate all the immunology that's going on in the Doherty. So I also thank everybody who's taken part in this discussion. I think it's been very helpful. It has been recorded and it will be available online, hopefully very soon. I'm not sure how that proceeds and where it will be, but we'll send you notifications, and it should be quite clear. It'll be somewhere on the Doherty website, I think. Is that correct, Mason? Yeah. Yeah, okay. So thanks, everybody. And yeah, stay safe, and we'll keep you in touch. Thank you.